Welcome, everybody. This is the ESIP Soil Ontology and Informatics Cluster. It is June 25th, 2025, and we are going to be hearing from Ashley Lang. Without further ado, go for it, Ashley.
Okay, well, thank you very much for being here and for listening. My name is Ashley Lang, and I'm a postdoc with the Forest Inventory and Analysis Program in the USDA Forest Service. And I work specifically in a group that focuses on forest carbon dynamics. But my background is in soil carbon and soil nutrient cycling. So I'm at the intersection there of forest, soil, carbon dynamics. And so that's what I focus on in this group. And so this is kind of a schematic of the way we think about forests as cyclers of carbon and all the different pools and fluxes of carbon within them. On our team, we kind of have individuals that focus on each of these pools. Harvested wood products being really important to a lot of folks, and then the primary focus being the above ground and below ground live biomass of the trees, because it is a tree focused data set, as you may expect from the Forest Service. But then there's also, if we're thinking about carbon, we can't ignore that there's a really important carbon storage process going on in the litter and soil components of the ecosystem. And so that's where I come in. When we think broadly about the US and all the forest land that we have nationwide, about 55% of the carbon in our forests is below ground. So we've got them, greater than half of it. And I'm really interested in, as you can see in this image on the left, it doesn't necessarily change quickly over time. This is a generally pretty stable pool, especially compared to the above ground live biomass pool. But it is nevertheless really large and also vulnerable to loss, as we know, with continuing changes in both climate and the way that we manage and treat our landscapes. And so it's a large and vulnerable pool. And the FIA program conducts the nationwide forest inventory, which is a tree inventory that is carried out every year. Some of the plots are sampled, and these plots cover the entire country, systematically positioned to represent all forest types, in relation to their abundance on the landscape. And primarily what is measured when crews go out to these sites is tree diameter and species and information about the health of those trees. So those are the core measurements that are taken. And then we have some geophysical parameters that we can count from each site. But every 16th plot is what we call part of a more intensive plot design. And in those plots, we also collect additional information about ecosystem properties. And one of the things that is collected in those plots are soil samples and also litter samples. And so the plot design is shown over here on the right. And then there's a map here of all the places where we have soil samples that we've collected, with the number of samples shown there in the color scheme. So most of them, we only have one visit. And in some regions of the country, we have managed to go out twice to collect samples from some of these sites, and then even 3 times in a few places. And you can see that the distribution of those samples is not perfectly even across the country, and some of it is because this is where the forests are relative to other ecosystem types. And some of it is logistics and, the ability of different research units to conduct the soil sampling, which may come down to things like funding or availability of field crews. So nevertheless, we have a lot of samples to work with. And it's the kind of thing where we have a really wide spatial distribution, and as a result, we have to make sacrifices in terms of the level of detail of the data. And so, for example, there's one core taken per plot, where in a biogeochemical study, you may typically want more than one. The samples go down to 20 centimeters depth, which is a wonderful thing to have for this many plots across the country, but other folks may want deeper samples for other specific data reasons. But long story short, we have a lot of numbers to work with. And I was really interested in understanding both the carbon stocks, the organic carbon stocks in soils in the upper 20 centimeters in these plots across the country, and then trying to understand a little bit more about what influences the variation in that number regionally across this map. Okay, so in the nationwide forest inventory, this is the way that litter and samples collected. You can see in this diagram, there's sort of a 25 centimeter radius circle that we collect litter from. And the depth of the litter is variable depending on the site. So it's you know, based on what's there. And it goes to the surface of the forest floor to the surface of the mineral soil. Once that sample is collected, and it's done sort of by hand, like a hand trowel, once that's done, the crews use an impact-driven hammer core, which is a super common device used in soil sampling, shown here with a slide hammer. And they collect two cores, one right on top of the other, each of which is about 10 centimeters deep, with a 5 centimeter radius. Okay. So those two cores are both mineral soil by definition, and they are 0 to 10 and 10 to 20 centimeters. So they're not necessarily horizon specific. And we don't have data on whether they're like A or B or anything like that. So once we have those cores and we run some basic analyses on them, we calculate the soil organic carbon content with this equation here at the bottom of the screen. And that's what I've been doing most of the time with these data, is calculating SOC stocks. Excuse me. And so what you can see here is that the SOC stock is calculated by taking the concentration of carbon in the soil in the core, that's the C sub I, multiplying it by the bulk density, BD sub I here, multiplying it by the thickness or the depth of that core, and then the UCF here is a unit conversion factor that scales that number from the units of the little individual core to things like kilograms per acre, things, you know, metrics like that, so we can look at things across space. Okay. However, the bulk density part of that equation is something that, you know, bulk density is used to translate numbers from concentration into stock. And it's because, you know, when you have a concentration of carbon in a small amount of soil, you still need to know how much soil there is per area, per volume in below ground, in order to then figure out the actual mass of all that carbon that you have present in that ecosystem. So bulk density does that for us, converting it from calculation into stock or mass per area. But there are different ways to calculate bulk density. And so this kind of equation, you know, concentration times bulk density times depth, it's not enough information really to tell you whether or not, like what you're actually getting when you calculate this SOC data or this SOC number. So you could get very different numbers by using bulk density, by using one of these particular bulk density calculations. And there's three that are commonly used. The first is what I call total bulk density in yellow here, which is the mass of a soil sample divided by the volume of a soil sample. That is like the standard definition of bulk density, but soil samples are not just comprised of soil. When you take that core out and you unpack it, try to pass it through a 2 millimeter sieve, a lot of material is not going to go through. In these samples, it's mostly not organic because we've removed the forest floor. So what we have in the greater than 2 millimeter fraction here is most of the time rocks, but also large roots. So we'll call those coarse fragments. And essentially the way that you treat coarse fragments in your soil sample has huge impacts on what you calculate as the soil bulk density, which then cascades to impact the number you get when you calculate the soil organic carbon stock. Okay, so total bulk density is if you were to take a soil core, rocks, roots, dirt, everything, weigh it, that's the mass you're using, dividing that by the volume of that coring device. So it's a standard number. It's in this case 181 cubic centimeters. And you have the mass of everything in there. The next method I'm calling the hybrid bulk density method because as you can see from this visual, it kind of involves the numerator of one method and the denominator of the other. So in this case, you take the mass of only the less than 2 millimeter fraction. So you're passing that sample through a sieve and you're only weighing what goes through the sieve. And that is the numerator of the bulk density equation. So soil mass divided by volume. And once again, this time we're using the volume of the entire core, 181 centimeters squared for FIA. So it includes the volume of those coarse fragments. And then the last method you can use is what's called fine bulk density. And in this case, you remove the coarse fragment mass from the sample mass, and you also remove the coarse fragment volume from the sample volume. This one is sometimes kind of frustrating, time consuming to do if you are directly taking these measurements in the lab, because what it means is you bring your core back, you pass it through the sieve, you weigh your soil, and then you have to take all of the material that didn't pass through the sieve and get the volume of that material, which if it's rocks, this is pretty straightforward. You can drop them into a graduated cylinder and see how much volume of water they displace. Roots sometimes don't behave so nicely when we try to do that. And so they float to the surface of the cylinder and they interfere with the meniscus and all sorts of problems. They also, because they're usually not very dense. And so they cause issues. But so in any case, calculating the fine soil bulk density accurately is very difficult to do and time consuming to do manually. You can also do something like a estimate of the volume by if for example you know that most of your coarse fragments are rock, you can use a standard rock density value to convert rock mass into rock volume, or even better, if you know what kind of rock it is, you can define the specific standard density of that rock type and calculate rock volume to subtract it out. But a lot of the time we have more than just rocks. So it's an imperfect thing to do, but it can be really useful for large data sets. Okay, so what I wanted when I first started with FIA, I was reading a lot of review papers that had come out already with had used the FIA soil carbon data. And I was reading the methods and seeing a lot of people talking about using the bulk density data to convert into the stocks, and nobody really described exactly how they calculated bulk density in these papers. So I was really curious to know, what the numbers might look like depending on the method that you use. And did it really matter if folks were including the coarse fragment mass and volume and in the different calculation methods that they were doing? And so I took all the data from across the nationwide inventory that we have soil samples for. And I calculated soil organic carbon stocks using each of these three bulk density methods. And the punchline is that it matters a lot which method you use in terms of how much carbon you would estimate is in the upper 20 centimeters of forest soils in the US. So Inconsistent treatment of coarse materials in soil bulk density calculations can lead to an overestimation of the SOC stock by 32%. And so if you were to, for example, leave the rocks in and multiply soil carbon content by the mass of the soil plus the mass of the rocks, which don't contain organic carbon, you would overestimate soil organic carbon by this amount. And it's about 13 micrograms per hectare, which is like 70% of the economy-wide CO2 emissions calculated from, I believe, 2022. So it's not an insignificant number, and it's a sort of a methodological challenge that unfortunately we can't ignore. Okay, so now that we know that generally it's going to have a big impact on the overall number, the overall estimate of forest soil carbon stocks across the country, I wanted to look at how it specifically varies with different ecosystem factors. So the first thing and the most obvious thing is that it's going to be the discrepancies between these methods are going to be larger in places that have more rock and root material in the samples. And so the difference is like 79% or something as you go from 0 to 100% coarse fragments by mass. And this is like, so you go from like 50 to, I don't know, some much larger number if you include rocks in the mass. And that's the total BD value here represented by the yellow line. And that makes sense because if you are including the rock mass, then the numerator of your bulk density equation is going to be higher and higher and higher, which means this is going to be a positive relationship across that coarse fraction gradient. And then with the other two methods, since we are taking the rock mass out, the value of this SOC stock estimate is going to go down, down, down as you increase your coarse fraction percent. Okay. Interesting ecological differences also sort of presented themselves in this study. So we found that in general, the SOC stocks were higher in hardwood forests, but more importantly, the differences in the SOC stock calculated with these different bulk density methods were more extreme in softwood stands. So if you were, if you were in a forest that was all things considered pretty similar, but one was hardwood and one was softwood, you would expect to see larger differences in the SOC stocks estimated with these different bulk density methods in softwood stands. This is primarily due to the fact that softwoods are more tolerant of rocky soil conditions generally. And so a lot of this is being driven by that, and also the fact that higher elevation soils tend to have higher rock content and also are commonly dominated by softwood tree species. And so this is important because softwoods, those softwood forests are in ecologically sensitive places like mountain tops and alpine sort of zones. And if we are trying to understand not just how much carbon is already present in those ecosystems, but how that will change over time, getting an accurate number on the current stock of carbon in those sensitive kind of places is super important. So here's another way to look at it. We split it into ecoregions. All these plots are forest plots, but they're located in sort of a larger landscape of other ecosystem types. And so forest plots in areas that are generally dominated by forest are in the top left. And then we have alpine forests that are in kind of a mosaic prairie situation in the bottom left, and then dryland forests on the bottom right. And so these show that the discrepancies between soil organic carbon stocks estimated with each method are higher in alpine zones and drylands, both of which are places where coarse fragment content tends to be higher. So pointing out that, once again, differences that are driven by coarse fragment content can kind of disproportionately impact certain places across the landscape. And alpine zones and drylands are also both ecologically sensitive places. And so it's important to nail down these numbers a little more. Okay, so when I think about these three bulk density methods, the total bulk density doesn't make a lot of sense to me at all. In terms of understanding nutrient and carbon stocks, you just never want to multiply the concentration of the elements that you're sampling or that you're measuring from the fine less than 2 millimeter fraction, but you wouldn't multiply that by anything that wasn't in the fine fraction. So including that mass generally is going to be an overestimate. So removing that one from our mind for a moment, I wanted to compare the two methods, hybrid BD and fine BD, that do in some way account for coarse fragments. They do it differently, and they're both completely valid for very specific purposes. And when I talk about bulk density with folks, I feel like I normally get about a 50-50 split of how people were taught to do it, either this hybrid method or the fine bulk density method, where you just use the fine soil mass and volume. I was taught the fine bulk density method, so I always believed that you have to put all your rocks into a graduated cylinder or else you don't know the bulk density. But there are a lot of reasons why you may not need that information depending on what your question is. So the hybrid method here on the left is completely fine and adequate for doing things like calculating the current pool size of carbon in a specific soil depth across an area. But it is subject to high uncertainty because you've got a, in the numerator of your equation, you have the mass of the fine soil alone, and in the denominator, you have the volume of the entire core. So that means you're going to have variability in rock content from core to core that is represented here by differences in the mass of the fine soil fraction. So because that difference is going to be pretty high from location to location, even within a small area, you need a lot of cores to get this number accurate. And in some, I think, there's power analyses that have been done to ask these kinds of questions, not necessarily for bulk density, but for other things like nutrient and carbon cycling. And a lot of times the number of cores that you actually need is just unreasonable and cannot be done. So this method, like I said, it's really, it does effectively give you the amount of carbon in the soil volume over a certain area, because in the numerator you have the soil mass and in the denominator you have the volume, which includes those coarse fragments. And that's a good thing because those coarse fragments do exist and they are displacing soil. And so, yeah, so you do end up with kind of the actual amount of soil carbon per area, with the understanding that the area is comprised of a matrix of both soil and non-soil material. Okay. The second one here is the fine bulk density method, which it's the mass of the fine soil divided by the volume of only the fine soil. And in this case, it's useful for understanding carbon fluxes and predicting change because it is more resilient to variability in coarse fragment content. And by that, I just mean it's essentially like the concentration of soil carbon per unit soil, because you have the mass of only the fine soil divided by the volume of only the fine soil. And so it doesn't matter how many rocks were in there because you're taking them out of all the parts of this equation. So sample to sample, regardless of coarse fragment content, you should get a pretty similar number within one small area when you use this method. Okay. So for this particular figure, I wanted to compare those two methods, the fine bulk density method and the hybrid bulk density method, and look at the difference in the carbon stock you would calculate with each of them. And then I specifically was interested in how that varied by forest type, because you would think that certain forest types are going to have maybe systematically different differences between those two methods based on the soil conditions where they commonly grow. And we did see that tended to be true, and it's kind of hard to tell from this figure, but generally the softwood species, which are represented by shades of green here, as we saw earlier, those ones had the highest discrepancies between the two methods that account for coarse fragments. And then in the purple shades, we have hardwood species. And so those were smaller differences, but for both tree types, you can see that the size of the circles tends to get larger as you go northward, and it also is higher at higher elevations. So these are places where it tends to be rockier, previously glaciated soils, shallower soils in mountainous regions, that kind of thing. So if we look at the differences in the SOC stock calculated with each method, this is a much easier way to see that the softwoods tend to have a greater difference, with the exception of southern pines, which are commonly grown in the southeastern US, and it's a plantation system. A lot of the times it has been, it was like previously ocean, in ancient times. And so these soils are old, not super rocky, and that's they are a little bit homogeneous in nature. And so that tends to be why southern pines are on the lower end of this diagram. Okay. So given that high elevation systems, softwood systems, and more northerly systems tend to have these discrepancies, I was curious to know whether in a place where these differences are most extreme, we might accidentally misinterpret influences on soil organic carbon content because of the method that was used to calculate bulk density. Like, what are the real world consequences of these differences between bulk density calculation methods? And I zoomed in on a place that met all these criteria, which is the state of Idaho. A lot of plots in Idaho are dominated by fir, either Douglas fir or true fir. And they grow in generally sort of similar elevations and things like that. So it felt like a good comparison to look at here, two softwoods. So if we were to look at soil carbon stocks in plots dominated by true fir and Douglas fir, and we use the hybrid bulk density method, we would get on this plot here, we would get what you see on the left rather, where the Douglas fir plots tend to have a significantly lower soil organic carbon stock compared to true fir. And then if we did the same analysis on the same plots, except we used only the fine soil mass and volume to calculate bulk density, that difference goes away. And the reason for this is that Douglas fir tend to grow in slightly rockier environments. And so their bulk density values are kind of, their bulk density values are going to be lower when we use the hybrid bulk density method because the rock volume is accounted for in that equation. So yes, there's less soil under Douglas fir compared to true fir, but it's because there's more rocks under Douglas fir generally. And it would be easy if you didn't really have this nuance in mind. It would be easy to use the hybrid bulk density method and to try to make some kind of statement or analysis about how Douglas fir is maybe producing less litter, or has some kind of impact on decomposition rates or something. We might be thinking about ecological differences between these species that aren't necessarily real. And so this is kind of a potential consequence, a potential misinterpretation of soil carbon data based on bulk density method. So if a user of the Forest Inventory and Analysis database was going to try to understand soil carbon stocks in forests across the US, they might want to look into how bulk density is calculated. And if you go to the FIA user guide, which has a description of all of the variables in the FIA data tables, you would see this statement at the top: bulk density. The soil bulk density calculated as weight per unit volume of soil. Weight of what? The soil, or the soil plus the coarse fragments, and then per unit volume of soil. It sounds like they mean just the fine soil, but it's not clear. And so if you dive a little deeper into other documentation in the FIA universe, you would find this paper from 2005, written by Catherine O'Neill and others, which describes it a little bit more in detail and tells you that bulk density is the of the mineral sample is the sample weight. That's what SW here means. Sub OD, which means oven dried. So it's the oven dried sample weight, all of it, including the rocks, divided by V, which is the volume of the core, a standard number, 181 centimeters, which means the rocks are included in that volume as well. So that is essentially the total bulk density method that I scratched out previously. It doesn't account for the content of coarse fragments in the mass of the equation or the volume. However, if you keep reading that paper, you find in another section, coarse fragment content is also measured, and it's separately provided in another column in the data frame, where you can get the percent of the total air-dried sample weight that is comprised of coarse fragments. And so with that, you can then correct the bulk density number to account for the mass of the coarse fragments. Volume is not available, so you would need to use one of those methods where you then take the mass and convert it into volume with a standard density number. But also noticing that this is using the air dry sample weight, whereas the bulk density equation uses the oven dry sample weight. So you do want to make a little adjustment there to account for the residual moisture in the sample. All this to say, it is not particularly recommended to just dive right into the data and use the column that's called bulk underscore density. And one may assume they know what that means, and then calculate a number that is actually a 32% overestimate of the national soil organic carbon stock in forests. And so we use these numbers to create models of forest soil carbon stock to depths of like a meter. In this case, this is an analysis that was done by my supervisor, Grant Domke. We can, the soils are only collected on about 1/16 of the plots, but we can model the depth, or we can model the carbon stocks to a depth of 1 meter in all the rest of the plots by sort of using these scaling procedures and biophysical parameters about the plots. But if we have, an imperfect measure of bulk density in the original empirical data, then we're just propagating this error up to the entire landscape and also down to the depth that we're trying to estimate when we do these models. So very important to get the best number possible that we can with the real data before we start introducing them to models. Okay, so the implications of this work are essentially that poor documentation and inconsistencies in the way that we calculate bulk density across studies, and also sometimes within them. And then also incorrect interpretations of what those bulk density values mean once they're calculated. And then the fact that we sometimes need to blend data sets together that are not always created with the same bulk density methods used. All these things lead to really potentially large inconsistencies and inaccuracies in our estimates of current soil organic carbon stocks, which makes it nearly impossible to then predict how they're going to change as we go into the future. Okay, so this paper, or this work has been published. It's in Biogeochemistry Letters. There are all the figures that you saw here are in that paper as well, in a little bit more compact form. But if you have questions about it that are not clear from the text, please get in touch. I would love to chat more about it. And thank you very much for your time today.
Yay! Thank you, Ashley. In the interest of time, I'm actually going to stop the recording and roll us into open discussions. So.